through our third line of defense. Okay, we had went through the first line of defense and the second line of defense. All right, so we went through our non-specific immunity that we talked about, right? Barrier defenses. We talked about cell-mediated defenses that are not specific to particular antigens, and then we talked about antigens before on Monday. All right. Today we have to finish immunity. All right, and what we're going to be talking about is our third line of defense, and specifically adaptive immunity. And this is specific responses to specific antigens. Okay, so we're going to be talking about a response to a particular pathogen, to Streptococcus bacteria instead of all bacteria. Okay, and our adaptive immune system, and we're going to get into this. It is actually controlled completely by the actions of cells. Okay, two types of cells in particular: B cells and T cells. Okay, B lymphocytes and T lymphocytes. So those lymphocytes that we found in the blood, okay, that we went through when we went through blood, and you saw the lymphocytes are present there, right? They're the second most numerous white blood cell in the blood, but they're really found in lots of places where. In what other system are these cells found? In the lymphatic system, especially lymph nodes and other lymphatic tissues. Okay? Macrophages are also part of this, but macrophages do not mediate the responses, but they are involved in signaling the beginning of the response. And you'll see that in a little bit. Okay? They are one of those antigen presenting cells that we had talked about last time. Right? We talked about antigen presentation. Right, in order to activate B cells or T cells. Okay, so a lymphocyte, those B cells and T cells, and why are they B cells and T cells? Where they mature, right? So B cells mature in the bone marrow, and T cells mature in the thymus. Okay, so those B cells and T cells, they undergo three distinct events in their lifetime, right? They have to be produced or formed, and they mature, so formation and maturation. Then they are activated when necessary, right? And then they elicit some type of response. Those three events are, you could talk about whatever you talk about, B cells or T cells, they will undergo this type of response, okay? So, we talk about B cells and T cells. The lymphatic structures, red bone marrow or the thymus, are where the T cells either, or let's say lymphocytes, are either produced and or mature. All lymphocytes originate from the bone marrow. Okay? They all originate from the bone marrow, red bone marrow. T cells specifically, they just mature in the thymus. Okay? And once they're formed and they're matured, now they become what's called immunocompetent. We'll talk about immunocompetence in a little bit, right? When they become immunocompetent is when they are signaled to begin activation. Okay? They can recognize, right, a foreign antigen, right? One specific foreign antigen. Again, we are talking about specificity. Okay? This is our specific immunity. Activation of our lymphocytes, right? When they run into, they experience, interact with, come in contact with a particular antigen, they become activated. Okay? So they're exposed to the antigen, become activated, and when they do that, they replicate and they form identical lymphocytes. This is called clonal expansion. Okay, clonal expansion. Yes, that's a C. We'll erase that for a second. Clonal expansion. And then from there, all right, when they recognize a particular antigen, each of those clones now will also be able to recognize a particular antigen. And now when they run into that antigen, they migrate to specific sites of infection or where their antigen can be found, and then they elicit some type of response. Okay? In T cells, that could be killing the cell. Okay? In B cells, that may be producing an antibody. Okay? Whatever it might be. Okay? And the T lymphocytes go to infection. B cells, they produce the antibody to that particular antigen and release it into the bloodstream. Okay? So let's go through this. Formation. We said that formation of these cells happens in the bone marrow. Okay? But now B cells, they stay in the bone marrow and they mature. Okay? T cells, they will migrate to the thymus and they will be selected. All right? There's a couple different ways of selection. Okay? Either positive selection or negative selection. All right? So thymus tissue will cut through and select out 95% of the cells that won't bind. Okay? So cells that go through the thymus, either they can bind, and positive, they have a positive influence. These cells are thymic. Okay? So we're going to pick out the right ones, and we're also going to let the wrong ones go through. So at the same time, positive and negative selection is ongoing. Okay? They either have the right antigen, the ones that do, and the ones that don't. Okay? It allows for complete, accurate selection. Okay? So your pre-T lymphocytes that go to the thymus. All right? We know that the thymus is a lymphatic structure. This is where they mature, and then they are released. And there's a couple different types of T lymphocytes. Okay? Your book the text goes into a very in-depth explanation of the T lymphocytes. I tried to simplify it as much as I could. Okay? And we're going to try to keep it at a simple part. You understand there's T regulatory cells, there's helper T cells, there's naive T cells, there's cytotoxic T cells. Well, what do those mean and how are they different? We'll talk about that in a little bit. Okay? So, now they become activated. Okay? Secondary lymphatic structures house both B cells and T cells. So we've talked about the lymph nodes, right? And lymphatic tissues come in through here and they have exposure to particular antigens. The spleen, the blood is filtered through the spleen. Again, possible exposure to particular antigens. The tonsils, where there's a opening to the outside surface. Many specific antigens can come in. That's, again, an exposure to potential antigens. Okay, so they're housed in your secondary lymphatic structures, right? And this is your site of activation and proliferation of these cells. By proliferation, what do I mean? What is meant by I can't talk today. Proliferation. Build up, they're going to grow, right? So we talked about clonal expansion. This is where we're talking about. Like, so proliferation is growth, right? Expansion is growth. They're going to grow in number, okay? And then the effector response. Helper T cells, right? Cytotoxic T cells. And then antibodies. You'll notice here, it actually says right here, plasma cell is a differentiated B lymphocyte that's been activated by a specific antigen and produces specific antibodies. On this side, what do you see? What type of cells are over here? You can kind of read right there. Taylor. What type? T. Okay, thank you. T cells over here. B cells over here. Okay. Cell mediated. Antibody mediated. You see the difference? B cells, they are responsible for antibody mediated responses. Okay? And T cells are responsible for cell mediated responses. Okay? So the B cells will produce antibodies that will do something to the particular antigens that they are reacting with. And the T cells, right, will recognize the particular antigen and will do something in response, but that response is a cellular response. It's not a protein and something that's produced by the B cell itself. Understand? Those are your two arms of adaptive immunity. Okay? You have a cell-mediated immunity and what we call antibody-mediated or also humoral immunity. All right? Here's another picture of it. Immature, naive lymphocytes are produced in the bone marrow. The thymus is where T cells go. B cells stay in the bone marrow. They both end up in your secondary lymphatic structures, lymph nodes, the spleen, other lymphoid tissues such as Peyer's patches, tonsils, etc. Okay? They're destined, right? So first, where they go, then they become immunocompetent. These are activation. They are potentially, right? They are ready, matured enough so that they can respond to particular antigens when they run into them or exposed to. Okay? And then eventually, number 3 down there, that's number 3 right here, they are activated. Okay? And in some cases, some of them become what we call memory cells. All right? How many of you, all of you, had a chickenpox vaccine at some point? Yes? Oh, I was like, yeah, I did. I remember that one. No, all of you did. You were all at the age where the chickenpox vaccine is prevalent. I think it started in like 1995. Okay? So you should anyone not have a chickenpox vaccine? Okay, I'm the only one. I had it twice. I had chickenpox twice. It was awesome. Okay? First time was great. Second time was even better. Just kidding. Okay? So, you have had it once, and now you remember that antigen, right? So you have what we call memory. We'll talk about that later. But those memory cells are some of those T cells and B cells that have been activated, and they store that memory so the next time you run into it, we don't have to go through the whole process again. Okay? So here's the beginning of all of this from start to finish. Okay? This is how, let's go through these cells. So here's a primary response. Okay, we have B cells that are present, immunocompetent. They run into a particular antigen. When they run into that particular antigen, they become activated. Okay? So antigen binding to receptor. All right, so we become activated. Now we begin, what's this called? Proliferation of what? Joe, proliferation of what? What's going on? We're talking about this. Proliferation of what? The cells are growing. What do you notice about these two cells right here? What do you notice about those two cells? Are they different than these cells? They're larger. What else? What do you notice about this cell, and this cell?
Different receptors.
They have different receptors. One of those receptors interacted with that particular antigen. Now, what do you recognize about these two cells? They're the same. So clonal expansion has begun. Okay? Why is that particular cell growing and not the green one or the light blue one? That's the one that the antigen is specific to. Okay? So it becomes activated and it starts to clonally expand. Okay? Those are B cells that have been activated. B cells are important for what type of mediated immunity? Call a antibody. Okay? So those activated B cells, some of them they divide, right? And most of them become what's called plasma cells. Plasma cells. right here. This one. Plasma cells. Plasma cells are the cells that produce antibodies. Okay? Antibodies are going to be specific to the particular antigen that this B cell just experienced. Okay? One of those cells became a memory B cell. It's going to stay. Okay? So a primary response is going to happen. You're going to produce antibody. And then over time, a secondary response will also occur. Okay? So later on, when you're exposed to that antigen again, that memory cell remembers it, recognizes that that's the antigen, and produces even more plasma cells. Okay? A larger response, more antibodies are produced. Okay? That's the basis of humoral antibody-mediated immunity. B cells are activated by a particular antigen, okay? And then they start this process. What do you notice about how did the B cell run into the antigen? Was there any intermediate between the antigen and the B cell? Yes or no? Zoe? No. Okay? That's one difference between cell-mediated and antibody-mediated. The B cell here can directly be exposed directly to an antigen and then start a response. A T cell cannot be directly run into an antigen. Okay? It has to be mediated through that antigen presentation that we talked about on. Okay? So when you have this primary response, look what happens. Okay? So how many of you have had a vaccine within the last 2 years that you remember? Okay? Whether it be hepatitis B or whatever else. Okay? How does it go? What happened? You walk in the doctor's office and say, hey, I need a vaccine. Okay, you get a shot, right? Then what? That's it? Walk out the door? If it's a flu vaccine, yeah, probably. But if it's a hepatitis B vaccine or something else, usually you have you have to get another one. Okay, you get a booster. Hepatitis B is actually 3 shots, right? The point of the series of shots, is the first time you're exposed to the antigen, you're going to develop an immune response. Okay? And it's going to go, and you're going to produce antibodies. Great, awesome. And maybe you also, and during that response, you produce a couple memory cells. But now you have that memory cell, and now I'm going to expose you to that antigen again. That secondary response is going to make a bigger response, okay? And you're going to produce even more antibodies. So let's look at the panel up here. On the left side, your left, you see the primary response, right? And the amount of antibodies produced in the blood. Okay? It goes up for a little bit, and then it goes down. Okay? But in the secondary response, it goes up really, really high, quickly. Okay? So a lot of antibodies are produced quickly. What do you notice about this primary response right here? About how long does the response when antibodies are produced take from the graph? About how many days? 4 to 5 days. Okay? We'll say IgGs, one particular immunoglobulin, gets produced after 5 days. What about after the secondary response? Way faster. So if we were to compare these two, a primary response takes more time and is not as robust as a secondary response. Okay? So the secondary response happens quicker, and that's a function of the fact you've already been exposed to that antigen and you have a memory cell present that can start this process faster. Okay? So humoral immunity, antibody-mediated, okay, and there's a subsequent response, memory involved. Okay? Humoral immunity can be classified in a couple different ways. Active and passive. Okay? And then we could also classify it as being natural or artificial. Okay? So the distinction between active and passive is whether, you answer the question, whether or not you particularly, or that person that we're talking about, is making and producing the antibodies themselves. Okay? So the answers, so producing antibodies. Yes, no. Okay? If yes, that's considered active. Okay? So I give you a vaccine. Are you going to produce the antibodies in response to that vaccine? I'm not producing them for you, am I? You're not getting antibodies from someone else, are you? No, you're not. So yes, you're actively producing them. Okay? So you're coming in contact with the antigen, whether it be a vaccine or from exposure to the outside itself. Passive means you are not actively producing the antibodies, you're getting them from somewhere else. Okay? And then we can further break that into natural or artificial. All right? Natural meaning, is it direct exposure to the antigen? Direct exposure to the antigen itself. Okay? Oh, this is artificial. All right? Artificial being exposure to some type of vaccine. Okay? So I had chicken pox twice. Okay? I have acquired immunity to this. What type of immunity? Did I actively produce the antibodies? Yes, I did. Did that happen naturally or artificially? Naturally. I was directly exposed to the antigen. All of you were exposed to the vaccine. So you're artificial. Okay? We can similarly do that for passive. Okay? So no production of memory cells in passive. You're getting, what this means is you're getting the antibodies from someone else or somewhere else. Okay? And this can also happen naturally or artificially. Okay? Give you an example. Mother to baby can pass antibodies through breast milk. Okay? And we can also cross the placenta. Okay? So, mother to child, is the child producing the antibodies themselves? No. So that is considered passive. Did it happen during a natural process? Yes. So that is natural passive immunity. Example of natural artificial immunity. Okay? My wife had never had chicken pox before. Okay? She was pregnant with my second daughter. I had to think about that, right? Second daughter, and she was teaching at the middle school she teaches at. Okay? And one of her students, who normally would be vaccinated against chickenpox, actually came down with chickenpox. My wife was in her third trimester. Okay? Now, why is this a big deal? If an adult gets chickenpox, it tends to be more serious. If a pregnant woman gets chickenpox, it can cause problems to the growing baby. Okay? So my wife, luckily, she got 6 weeks off from work. She got dismissed. Medically, I can't be there. I'm out. She was like, summer started early for me in the second week in May. Cool. Awesome for her. But as that is one precaution they took, the second precaution was they gave my wife a shot, an immunoglobulin shot of antibodies to the chickenpox virus. Did my wife produce the antibodies? No. Did she get them in through a natural process? No. So now she has immunity against chickenpox for a period of time. What was it? How do you classify it? Artificial passive immunity. Okay? One of the big distinctions between active and passive is not long term. It's short term. Okay? So children have to be vaccinated even though their mom may be exposed to particular antigens when breastfeeding. Okay? Because there's no memory that goes with it. Okay? So passive immunity has no memory cells, but active immunity does. That's the basis for vaccinations. That's the basis for active immunity. Get things twice. This is the structure of an antibody. So the things we're talking about. Antibodies are all the same. They have a shared, what we call a constant region of an amino acid structure, protein structure. All right, and then there's this important part, the variable region. Okay? The variable region is important because that allows one antibody to be different and respond and bind to a different antigen. Okay? So the variable region. And they're in this different structure, the heavy chain, light chain. If you want to be an immunologist, go study it. I don't care that you know it right now. Do you know what antibodies are? Yes? Do you know we're going to know what they do in a second? Okay? But I'm not concerned that you know the protein sequence. You know where the variable region is, and that allows them to react to different antigens. Clear? Okay. Now they come in different flavors per se, right? And they come in with there's immunoglobulin M, A, D, G, and E. And they do different things based upon who's producing them and in what response to different antigens. Okay, and I wrote down a couple of these. These are your 5 major classes. Okay, IgM what we call fixed complement. IgA is found mainly in mucus. Okay, IgD, important in the activation of B cells. You see that in a second. And then IgG can cross the placenta, right? And fixed complement and IgE are involved in allergies. All right, so depending upon the type of activation can mean the different type of immunoglobulin that's produced. Okay? And then it can also mean the different type of effector response. Okay? So we talked about activation of B cells. We talked about what they're going to do. Really, this is their effector response. They're going to produce antibodies. And these antibodies can act in a couple different ways. Okay? You don't need that one. We just go here. Okay? Antibodies can neutralize certain things. Okay? Example, viruses. So what they do is they take whatever particular antigen they're going to bind with, and they're going to take it out of prevent it from doing anything bad. Okay? Example, get bit by a coral snake in Florida. My wife used to work at the veterinary clinic, the vet hospital at the University of Florida. Right? Plenty of dogs would come in with a coral snake bite. Immediately what they do is they give them a shot of antivenom. Okay? The antivenom neutralizes the toxin. The antivenom is antibodies to that venom, that protein, and it prevents the toxin from binding and causing problems, right, throughout the body. All right? So antivenom, rattlesnake venom, stuff like that, that is a neutralization reaction. You're going to stop it from doing what it would do normally. Okay? Agglutination. You've seen this. You all did this in lab. Right? What happened when you mixed an anti-A antibody with A-type blood? It clumped. Okay? That's what agglutination is. All right? Antibody recognizes its antigen. It clumps together. Okay? Precipitation is somewhat similar. All right? In this case, it doesn't necessarily clump together, but the antibody binds to its it's soluble protein and then takes it out of solution and makes a insoluble complex. Somewhat similar to clumping. Right? These two are somewhat similar. Right? But here it's not producing a large clump. They're just coming out of solution. Okay? Those three. There's a couple others. So that's the antibody running into its antigen and performing a function on its own. Okay? Otherwise, there's also a secondary function that can occur. The antibody can stimulate something else. Okay? Complement fixation. All right? Complement is what? Do you remember what complement is? When did we talk about complement? Complement is a whole bunch of proteins that are present in the plasma of blood. Okay? And when that bunch of proteins becomes activated, it performs a nonspecific immune reaction. Okay? I have to sneeze. Here, we're utilizing the complement proteins that are present and starting a specific reaction by using the antibody that's to a specific antigen. Okay? So antibody runs into the antigen, in this particular one, so let's say it's an IgM, because it can fix complement. It takes complement out of solution on the other end, and now complement proteins will be activated and will break down and lyse that cell. Okay? But it can fix and start that reaction in a specific process. Okay? Another one, opsonization. Okay? In this case, antibody recognizes its antigen, okay, and it attracts a macrophage or a phagocyte to come and take this whole complex in. Okay? Maybe otherwise, without the antibody, the macrophage, the phagocyte, doesn't recognize this particular cell, but the antibody makes it. Okay? Makes that unique. And then also, finally, the activation of natural killer cells. The antibody binds to its particular antigen and then attracts the natural killer cell to kill this particular cell. Okay? So, complement, phagocytes, and natural killer cells are all examples of non-specific immunity, non-specific lines of defense that, because they are through antibodies, they're being activated through antibodies, have a specific response. Right? They bridge the gap of specificity in that case because you're using the antibody to a particular specific antigen. Questions on antibodies, what they do, and humoral immunity. Okay? Because now I'm going to blow your mind with cell-mediated immunity. Okay? Cell-mediated immunity means what? Are antibodies involved? No. Okay? What type of cells are we going to be particularly looking at? T cells. Okay? So this is, again, still specific, but it requires the presence or the activation by an antigen, but that antigen in this case, unlike B cells, needs to be presented by another cell, right? Whether it be an antigen-presenting cell, a dendritic cell, a macrophage, whatever it might be. Okay? When that happens, T cells then will recognize what that is, and they need to recognize what's you and what's not you. Okay? So they have double recognition possibilities. They recognize what is self versus non-self. Okay? And now these cells can also, once activated, they can talk to B cells and activate them. Okay? When we talked about humoral immunity, there wasn't any, hey, B cells aren't going to activate T cells, right? That line of communication doesn't exist. Okay? But T cells can be activated and they can stimulate B cells to produce antibodies. All right? So here's a dendritic cell that has phagocytose, phagocytose, whatever you want to call it. It ate the particular antigen, it processed it, and then presented it on the outside. So right here you have antigen presentation. Okay? That will then, that antigen complex binds, interacts with a T cell, in this case a helper T cell, and will recognize that particular antigen, and then will release cytokines, small molecules that will help activate cytotoxic T cells and B cells in response to that particular antigen. Okay? And then you get both arms of the adaptive immunity that are activated, both cell-mediated and humoral immunity mediated that are now activated. What does this do? We've been kind of separating these out, talking about humoral and cell-mediated. Are they separate? Casey, no. They work together. Okay, absolutely they work together. Okay? It doesn't matter if which one starts the process, there's going to be some type of overlap. Right? They're going to work together and they work simultaneously. Okay? So T cells, when activated, they will undergo clonal expansion. All right? And you have cytotoxic killer T cells, we call. Okay? These T cells, they kill other infected cells. So it will recognize, this T cell will recognize the infected cell as you, but it's not healthy you cell. All right? And it will insert a toxic chemical into the cell's membrane, something we call perforin. That protein gets embedded into the membrane and then causes the cell to explode. Okay? Other helper T cells, they're the ones that will interact with B cells to start that process. Okay? So here's the effector response of cytotoxic T cells. All right, activated cytotoxic T cell runs into this effector cell. You get recognition of both self and the fact that it's abnormal. Release of perforin. Perforin embeds into the cell membrane, okay, and releases this other granzyme. It's an enzyme that goes into the cell and basically digests the cell from the inside. Cell undergoes apoptosis. All right, controlled cell death. Right? Have any of you ever seen a controlled demolition of a particular structure, a building? Okay, in a city or a urban area where there are large buildings and they need to demolish one, they'll go inside the structure and detonate explosives to systematically allow the structure to fall on its imprint. Imagine that's the same thing that's happening, right? We're going inside, granzymes are going in, breaking stuff down on the inside so the cell kind of implodes on itself. Okay, here's another picture of that. Perforin molecules are inserted, they form a pore, granzymes are released into the cell, and then breakdown of the cell occurs. Okay? Other T cells, regulatory T cells. All right? Regulatory, T-regs, as they're called. They regulate the activity of other cells. All right? So they're going to release chemicals that will suppress the activity of T cells and B cells. All right? They're going to stop the immune response. They help regulate the immune response. All right? And then a few members of these T cells are memory cells. So you both have memory B cells and memory T cells. But memory B cells are far more numerous. Okay, so cytotoxic T cells, helper T cells, and regulatory T cells. 3 types of T cells that are important. Cytotoxic T cells will kill other infected cells. Regulatory cells will suppress and control the immune response, and helper T cells help to activate the B cells when they are activated. All right, so this is I love this picture, okay, because it kind of summarizes exactly what's happening when you're exposed to an antigen with the 2 things coming together, the 2 arms coming together. Okay? On the left, you have the humoral antibody-mediated response. On the right, under the orange color, you have the cell-mediated. Okay? Antigen first exposure, engulfed by a macrophage, becomes an antigen-presenting cell, and that stimulates, that stimulates a helper T cell. Okay? But that antigen can also go directly and run into a B cell, and that antigen can also be displayed by infected cells to start a cytotoxic T cell response. Then you have your helper T cells that can go further stimulate a B cell to the right, to the left, or stimulate your cytotoxic T cells to the right. Okay? Some of those become memory T cells, which will further stimulate both memory B cells and memory T cells. And then all the way over to the right, you have your plasma cells that produce antibodies. All right, and then your active cytotoxic T cells. And what do you end up with? They defend against extracellular pathogens, all right, by binding, whether it be binding antigens or by killing off cells. All right. One of the important cytokines that is present in how the helper T cells stimulate this is a cytokine called interleukin 2, IL-2 and IL-12. Okay, 2 important cytokines that regulate these processes. All right, they're produced by helper T cells, and they will stimulate cytotoxic T cells or B cells, depending upon what cells are present at that time. Okay, so in general, formation. We know what formation of lymphocytes. They both are formed in the bone marrow. They either mature in the thymus, in the case of T lymphocytes, or in the bone marrow in case of B lymphocytes. All right, once they become mature, they migrate to different areas of the lymphatic system, and they can be activated upon exposure to a particular antigen. So activation occurs, you get clonal selection based upon what particular antigen has been exposed. IL-2, important, right? In this process, here's an antigen presenting cell producing MH class 1 or 2, helper T cell, cytotoxic T cell, right? And here's a helper T cell that now activates a B lymphocyte to produce antibodies. All right, so these are the 3 pictures that are in your text, right? I like that one that flow chart that brings it all together. You can just kind of follow it rather than the 3 separate, right? And then your effector response, 5 types of responses by antibody, whether they be clumping, neutralization or precipitation, or by activating a nonspecific response, natural killer cells, phagocytosis, or complement fixation, or cell-mediated cytokines are released by helper T cells, and they start the production of cytotoxic T cells to kill off infected cells. I tried to simplify this as much as possible. Okay? Read the book, but understand the main ideas. I will ask questions on the exam that will test whether or not you understand the main ideas. I'm not going to ask you if a regulator or a T reg cell exposes CD4 or CD8. I don't care. do you know what a T regulator or a regulatory T cell does? That's the main important. Okay, that's the main idea. Okay, and then the last little bit within your book, they also talk about clinical views, hypersensitivities, and immunodeficiencies. All right, they also talk about vaccinations, but I think we covered that to a point. Hypersensitivities, all right, they are an abnormal or exaggerated response. Hyper means over, sensitive. You're oversensitive to a particular antigen. Okay, another also known as allergies. Okay, they could be acute, occur pretty quickly, subacute, 1 to 3 hours, right? And both involve some humoral immunity. They're going to activate and produce antibodies, or you could have a delayed, which occur 1 to 3 days afterwards, right? And produce cell immunity. Okay? Acute hypersensitivity is an allergy, right? The antigen you're exposed to is an allergen, a non-infectious substance such as shellfish, bee sting, peanuts, pollen, etc. Latex, right? And this may be from mild to severe. I already have some stuffiness from pine tree pollen, right? Come on, it's your fault, right? Runny nose, watery eyes. I'm allergic to Colin and all his friends. Okay, red welts, itchy skin. Eventually, more severe, could lead to vomiting, diarrhea, right? And even anaphylaxis, right? Shock, closure of constriction of airways, which cause problems. And then immunodeficiency, right? HIV and AIDS, acquired immunodeficiency syndrome, right? That is AIDS. It's a life-threatening condition, which is more of a chronic condition now. Result of HIV virus, which infects and destroys helper T cells. Where was the helper T cell in that picture I showed you? In the middle, right? It's in the middle. So that helper T cell in the middle helps activate and talk between the two parts of this adaptive immunity. Okay? So if they start to die, you end up with your immune response is lessened. Okay? And it can be transmitted by sexual intercourse, needle sharing, blood, exposure to blood, prevention through safe sex. And then this becomes AIDS when your helper T lymphocytes below a certain level. Right? So when you have that, you have opportunistic infections. Right? One of the signs of someone who has AIDS is what we call Kaposi sarcoma, like these red lesions that are on their body. Those lesions are cancerous, abnormal cells that are growing that normally in a normal immune system, in a sufficient immune system, would be recognized and killed by cytotoxic T cells and eliminated from the body. But when you don't have the helper T cells to activate that cytotoxic T cell, you can't eliminate them. They accumulate on the skin and form lesions, right? Because you have a opportunistic infection that will also be around. And then most of the time, this is fatal, usually to complications to some type of pneumonia or other opportunistic infection. Okay? So no cure, treatments alleviate symptoms and help prevent the spread. Okay, so we covered, that's it. We finished immunity. I smushed in 2 lectures into one. Awesome for me. Okay? On Monday, we will start the respiratory system. Okay? Have a fantastic day. Weekend.